Da vi klare med neste presentasjon, som dessverre ikke kunne være her i dag. Karl Trygve Kalleberg skal snakke om Nanopore sequencing. Det en video som han har spilt inn for oss, og så det mulig å stille han spørsmål remotely når den videoen ferdig. Så da det over til han egentlig.
Hi, I'm Karl Trygve Kalleberg, one of the Hackeriet regulars. Today I'm going to venture out on a bit of a limb and talk about something in the cross-section between biology and computing. The topic of today is DNA sequencing using nanopore technology. I've tried to make this talk accessible to newbies, while hopefully also somewhat entertaining to people familiar with the topic. So I wanted to talk about this one because I think there are a lot of similarities between biology today and computing in the 70s. Slowly but surely, biology is now becoming a software problem. And what's more, a lot of the hardware needed to read and write biology is becoming available to individuals, not just large institutions. So if you're into reverse engineering like myself, picking apart the genetic code is actually quite rewarding. And today, I'll try to explain to you how we can use the DNA sequencer in the same way that we might use a camera scanner, that is, the digitized DNA, so that we can analyze it on the computer. This talk is going to be rather basic, and I'll try to explain the broad strokes, but I'll leave in some details as well. DNA sequencing started in the 70s when people used to do things entirely by hand, not unlike computing in the abacus. Then, in the late 90s, the process became automated, and somebody in marketing decided to call this next generation sequencing, which was clearly the most future-proof name ever. NGS is kind of like mainframes of sequencing. The NGS machines are expensive, but also very high throughput. They live in core facilities and are run by people with many academic titles who wear white lab coats. The latest generation of DNA sequencing, often called fourth generation sequencing, because why bother counting, is more like the microcomputer revolution back in the 70s, 80s. So let's have a DNA sequencer on every desk, why don't we? Another similarity between the mainframe era and the desktop era is the approach to the software required to run these beasts. A lot, if not most, of the production software for the NGS machines are very closed source and mainly run on Windows. While the nanopore technology is very much a product of its time too, and practically all of its software is fully open source. The rest of this talk is going to follow a rather straightforward path. First, I'll talk about how the biology works, a small recap there. Then I'll talk about the nanopore hardware itself and the wetware, and by wetware I mean some of the organic components on the chips. Then we'll cover how to do the data analysis, at least at the very high level. I'll show some examples of applications and application areas, and then a few pointers for those of you who want to start yourself. And then I'll wrap up. So let's start with a biology recap. The DNA is an information storage molecule. Two people with the same DNA will look and act very, very similar, as anyone who's met identical twins will know. If you take a human zygote, that is the first cell after the sperm and egg cell fuses together, and replace the DNA, it will become an entirely different person. Arguably, the most important thing about us is our genes. They are the only thing that may survive us, at least as long as we get kids. And some people claim that our primary purpose even is to be just a vessel for our genes. Our DNA is split into chromosomes. Think of it as splitting a large string into multiple files. We have 23 pairs of chromosomes. For each pair, one copy came from your mother, the other from your father. Usually, one of the copies is active at any one time, and there are mutable configuration settings for which one is off and which one is on. And this gets us back to identical twins, actually. So while identical twins have the same DNA sequence, they don't have the same configuration settings, so they will never look exactly the same. The DNA itself, as we might remember, lives in the core, the nucleus of the cell. And famously, it consists of two strings that are zipped together and curled up in a helix, the double helix. This can look like a ladder, if you will, and each rung of the ladder consists of two bases, a so-called base pair. Each base here is sometimes also called a nucleotide. These bases are abbreviated A, C, T, and G for their molecular names: adenine, cytosine, thymine, guanine. And the job of a DNA sequencer is to read out this string of bases in some way. There are about 4 gigabases, 4 billion bases in every human cell. And our cells are fairly small, so this is an extremely space-efficient storage mechanism. And remember, it's not just the primary DNA that contains information here, but also the bundle configuration that I spoke of too. So for decades, some mad scientists have been trying to make DNA hard drives, or wet drives, I guess. And just like the flying car, these are just around the corner. Okay, so that was a very quick crash course into molecular biology. The main message is this: there is a string of A's, T's, C's, and G's that we want to read out, because that string is at the core of who we are and how we work at a molecular level. The ultimate reversing challenge, if you will. So, back to the camera scanner analogy. In 2014, the company Oxford Nanopore Technologies released this QT here. It's a handheld DNA sequencer that plugs into your USB port. They follow the inkjet pricing model, where they essentially give away the sequencer for free, and then they sell you flow cells, cartridges, that you spend as you read DNA. The biggest flow cell you can buy for this thing costs about $500 to $900, depending on volume, and it can produce about 50 gigabases of data before it runs out. By comparison, the human genome is about 4 gigabases, but sadly, it turns out that you can't just snap a single picture and get it right the first time. If you want to get good quality data, you have to be prepared to reread the DNA about 20 to 30 times. The appeal of this thing is that you can take it with you anywhere. It has truly made sequencing easy, and by now it's been used on all continents on our planet. But as I said, DNA sequencing has been around for a long time, so what's the unique part about this particular sequencer that enables this extreme versatility? In a word, that's the nanopore. This is a 3D rendering of the nanopore. The dark blue blob in the middle is the nanopore itself. It's embedded into a membrane, and the membrane separates 2 chambers, kind of like a membrane in a battery. The DNA molecule is designed to slip through the pore from one side to the other. You start out with DNA only being on one chamber and then moving on to the other chamber. And there's a motor protein that controls the speed of the DNA molecule. You don't want the DNA molecule to slip through the pore too quickly, because you want to measure a change in current that happens as the DNA molecule slips through this thing. And if you read out that current change correctly, you can actually detect which specific DNA sequence it was that slipped through, because each of these spaces induce a slightly different current in the readout. So I'm gonna go back and explain this a couple of more times, because I think it's super interesting and also a little bit tricky, perhaps. But the principle is actually quite simple. Again, this is kind of a setup of a single pore, if you will, kind of where they started back in the day. So you have 2 chambers. They're both filled with water, fluid, with ions, basically electrolytes, right? In that sense, it's kind of like a battery. And the chambers are separated by a membrane, and the membrane has a tiny channel in it, and the channel is the nanopore. So there is a voltage difference that you induce between those 2 chambers, and once you induce that voltage difference, then electrolytes start moving through the pores, and that leads to a current. So that gives you kind of like a constant current through that pore. From time to time, however, DNA also tends to slip through, just because of concentration differences, and also because DNA is a charged molecule. And whenever the DNA molecule slips through the pore, it transiently blocks this pore, And that, of course, would lead to changes in the current of the normal electrolytes that will go through there. And then the quote-unquote only thing you need to do is to plug in a digital ammeter, and that gives you the readout, and then you plug in your own network on top of that readout, and then we have a DNA sequencer. How easy could it be, right? So another way to look at this is kind of like the same kind of thing. It's a different figure, but basically the movement of the DNA through the pore, that leads to a change in the measured voltage. So you have this thin pore-infused membrane, separates two chambers, one called the cis chamber and one called the trans chamber, and then you add a voltage difference, and the voltage difference, of course, is controllable, but that's tweaked by the system. And then, as I said, for the most part, there will be no DNA in the pore, and then there's going to be a current going through that pore. But from time to time, as the DNA blocks the pore, we can read the corresponding changes in the current as each of those bases go through. And here you see the bases in the different colors, and the conceptual model is that sort of each of them will lead, when they are in the pore, in the middle of the pore, they will lead to a different current than the other bases. So you might ask, how did I find that pore then? What was the pore made of? And the pore they stole from nature, actually. They stole them from bacteria for the most part, and they tried a lot of different pores, and here is a list of some of the ones that sort of start out early. And these pores, they exist in nature. We have them in practically all of our cells, and so does bacteria, right? And once they found a suitable pore that was close enough to what they wanted to use, they started gene modifying the genes of that pore. That pore came from bacteria, so that they could iterate and just adjust that pore to match what they really wanted. So the pore that Oxford Nanopore is using is a derivative of a pore protein called alpha hemolysin, or also alpha toxin. Perhaps it's the first and most commonly used biological pore by now. And it's actually a toxic substance secreted by the bacterium Staph aureus. And Staph aureus is a skin bacteria. You find it in practically all of us have it on our skin from time to time. And this is a fairly sort of important disease inducing bacteria in our lives. So in one sense, it's good that we found a more pleasant use of that pore for ourselves. So the once that pore is in place, and then when you have the ammeter there, then the currency change. This is kind of like an example of an early readout that came from this kind of technology, right? So here you see some steps, and those steps, for the most part, correspond to the various bases that exist in the pore at that time. And then, depending on how quickly the DNA goes through the pore, then you will see quick changes over time, or if it moves through the pore at a slow pace, remember this is what the motor protein is for, then you will get a better sort of temporal resolution. You have more milliseconds, if you will, to detect the signal, and that's the main purpose of the motor protein. So now, equipped with sort of a single nanopore that we talked about, then what of course they did is to sort of try to parallelize this, right? So once a single nanopore was sort of working, they put it together in a big array. So if you look at it from the side here, you will have multiple different pores, and green blue parts at the top, those are where the pores are, and then there's a lot of electronic circuits around that to be able to read out the current change, and also to be able to adjust the difference in voltage between the chambers and a bunch of other things. And one of the important parts of this setup that they've been doing is that they wanted to make this fairly modular, right? Because they wanted to be able to switch out the pores separately from switching out the other components, so that they can iterate quickly and make sure that this sort of system could evolve over time. And they mostly succeeded in that. It's actually now possible to switch out the pore protein and the chemistry somewhat differently, sort of separately from the rest of the system. You don't have to upgrade the entire machine if you want to upgrade the pore. So in the end then, this is kind of what's happening inside of this image that we see here, the machine that is here. The flow cell, the black part here, has about 512 channels, sort of pores, per flow cell. So that gives you the ability to read sort of 512 DNA molecules in parallel. The ammeter has a sampling rate of about 4 kilohertz, so it's not a lot actually. And the multiprotein is adjusted typically. The combination of the multiprotein and the voltage difference, I should say, is adjusted so that the DNA generally moves through the pore at around 400 bases per second. And if you multiply that up a little bit, because the flow cell expires over time, the pores stop working, they're biological after all, things get stuck, they get destroyed. So typically you will be able to run this thing for about, once you start it and put it on, it will sort of slowly, the DNA will diffuse from one chamber to the other, right? But after 48 hours, it usually doesn't work so well anymore. Then it usually stops. And that gives you, with all the loss and everything, about sort of the ability for a single flow cell to produce about 50 gigabases before it runs out. And it is actually possible to wash and reset this flow cell. It works a little worse the second time, but you can run it a couple of times and then it's spent. You have to send it back to the company and they can actually replenish it. So one important thing to mention at this point is DNA fragmentation, because I guess in your mind now you think of DNA as just one very long molecule, and as long as we read it in a single pore, then everything is good. But that's unfortunately not what's happened, right? So the DNA molecule is already split into chromosomes, as I said, but also when the sample is prepared for sequencing, these chromosomes are fragmented. They become fragmented. So the DNA sequencer will only see a bunch of shorter and some longer DNA fragments. And these fragments are usually in the hundreds to thousands of bases long. And each of these fragments will then result in their own read, the signal chunk, the squiggle at the bottom. So the resulting file from DNA sequencer, this DNA sequencer at least, is essentially a long list or a big file containing these signal chunks, these squiggles. Some are longer than others, and the length of each of these squiggles depends or is proportional to the length of the fragment that they were sequenced. So this kind of the machine, the hardware itself, leaves us with this sort of huge puzzle then to solve, because we don't know how to order these things and how they fit together. We just have a bunch of random, seemingly random reads that come out of this machine. So this leads us to the next thing, right? Because the Nanopore DNA sequencer that I've explained to you is almost nothing but a glorified ammeter. I mean, of course, there's a lot of tricky stuff, especially the biology electronics combination there. That's highly non-trivial. But the only thing it produces is a squiggle. And the squiggle produced by the ammeter is not exactly what we wanted, right? We wanted the actual sequence of the A's, C's, T's, and G's. So this is, of course, then where it turns into a software problem. So the beginning here, of course, is what we talked about. The DNA goes through the pore and we get out this squiggle, this signal, the raw data acquisition, as it were. And then immediately the next step is to try to interpret that signal and produce the A's, C's, T's, and G's, the basis from that. And if we do that naively, then we'll end up with a short fragment of these letters for each squiggle. And then usually the next step at that point is to do quality control to try to determine how certain we are that we managed to affix the correct A, C, T, and G to the squiggle that we got out. But once we are sort of reasonably certain that we have an idea of how accurate each of those conversions were, then we move on to the primary data analysis. And this can be multiple different things. It could be basically trying to find specific sequences that are related to cancer. That's one application. That's sort of a classification application, for example. Or it might be that we try to start assembling these things in a sense, or at this point I think we usually start aligning these things to see if we can sort of find overlapping things and try to puzzle them together in a little bit of a way. And then if we actually want to have the whole genome, that's not often you need that, but many cases you do, then you move on to assembly, and then the point is sort of to take all these fragments and trying to put them together in a long list and try to recreate the genome from end to end itself. And that gives you the ability then, once you've done that, to look for various kinds of mutations that are sort of population related, eyes, genes that are relevant to people who are healthy or unhealthy. That's kind of like usually where this goes. And you can do the same thing for gene expression on RNA, not DNA. And so the primary data analysis and the secondary data analysis steps, those are highly dependent on the application you want to run here. And we'll get back to that a little bit later. And then, of course, once you've done these things, it's usually about emitting a report of some sense to human consumption. And I suppose one thing that's personally, I think, is sort of super appealing with this particular technology here is that practically all of the tooling for the ONT sequencers, they're open source, from the base calling to the very left-hand side, all the way through the pipeline to the right-hand side. Mostly, practically all of it runs on Linux, Docker, these kinds of things. So, going back now sort of to the main thing that comes out of the sequencer hardware is this current versus time graph, and this is the raw sensor signal. But in addition, there's also a bunch of useful metadata which is used when doing base calling. And base calling, we'll get back to that, but basically base calling is the name of the thing where we go from the squiggle to the letter, from the squiggle to the sequence A's, C's, T's, and G's. And the data that comes out of the sequencer used to be in a proprietary format called FAST5. Not super proprietary, they use an open format called HDF5. And there's a definition of it, but they're proprietary in the sense that nobody else or the other sequencing companies use this format. Now they've invented a new format called POD5 that they invented this year, another proprietary format, but proprietary in the sense that they're the only ones using it. This is based entirely on an Apache library called Arrow, which is gaining traction in all sorts of data science applications these days. And these two formats are functionally equivalent. They contain the raw sensor signal and the metadata, and they can also actually contain the DNA sequence itself once it has been base called, so it's possible to add a new record in there to have that information. But the main point about the POD5 format is that it's a lot more efficient to write, especially, and also to some extent to read, because it's much more linearized than the FAST5 format used to be. Anyway, at this point, I suppose I've been throwing around some terminology at you, and without sort of fully explaining all of it, so I'll make an attempt at that. This is, in all of the fields of biology, informatics, and bioinformatics, everything tends to have at least two names. I guess for many fields, this sort of is the case, but I'll try to be somewhat consistent in this talk as much as I can. And base calling is, as I said, when you go from the raw squiggly line signal to the specific letter that has to do with the squiggle. This is where you sort of assign the nucleobases, the nucleotides, to the currents. And then another thing you might want to do here in the sort of the data processing is alignment. So if you have two sequences of A's, C's, T's, and G's, like the DNA sequence, you might want to see if there's an overlap, and you might want to see if they fit together somehow, kind of like the basic elements of starting to puzzling it together. And if your goal is to try to assemble all of these things in a sense to a full genome, that becomes more of an assembly question. It's more a terminology difference between the alignment and assembly, but there is an important distinction here that I'll get back to. But the point about the assembly is that we want to reconstruct the original sequence. So if you put in a whole genome as the original sequence, then that's what you want to reconstruct. Fragment I've already used, that's kind of just a fragment of the DNA, like so it's broken off, and then you have some kind of like a string that's been chopped up in multiple pieces, and each of these pieces is then a fragment. And the read sort of corresponds one-to-one to a fragment. So if you put a fragment through the DNA sequencer, and then you do base calling, you will get a read that matches that fragment. And it's called a read. That's just the name of the base called output. Now, let's sort of take one step back now, because there have been many details. But what we wanted to do here, let's just revisit the problem statement. We want to take a biological sample from somewhere, a rabbit, a human, a plant, something. And then we somehow get the DNA, or even RNA, but I'm talking about DNA here for now. We do sample preparation, and we do the sequencing. And then we do the base calling that I talked about. That gives us the DNA sequence from the sample. And when we do this, I said we could add that to the FASTQ file, but usually you get out a different type of file called the FASTQ file, because that's the standard format in the field. And the FASTQ files are very simple in computer science speak, at least they're text files. Each of them, a FASTQ file, will contain thousands of records, and each record here is sort of 4 lines. The first line is the label, and the format of the label depends on the DNA sequencer manufacturer. So Nanopore has its own types of labels, and the Illumina machines have their type of labels, etc. Then there is the DNA sequence that has been base called. So this is the sequence that we believe came from the fragment that we sequenced for this particular fragment. So each record here is for a given fragment, I should say, just to be upfront about that. So then we get the sequence for the fragment. There's a separator, a plus, that's fixed there from the format. And then there is a quality line that basically for each of those bases on line number 2, the CCGT here, there is an estimation of how sure we are that that's correct, using a format called a rating scale called a Q-score that I'll get back to. And that's encoded then as some ASCII characters, basically. It's kind of like an encoding of a scale as ASCII characters. So this is essentially the problem statement that we're looking at here is the problem statement for the base caller. Go to from squiggly lines to this particular format here. And then that's exactly a job where that's not so easy to solve, because as it turns out, there are subtle differences between the current levels of each of those base pairs. And they're subtle enough that you can't just sort of do an if condition here, unfortunately. It's not a lookup table kind of problem. And this is an area of active research for nanopores. And sort of the current workhorse for this kind of problem is neural networks, basically. This is what they're using for these kinds of things. And every few months, a new neural network comes out that's a little bit more accurate than the previous one. And at the moment, the latest and greatest currently lives in the Dorado repository on GitHub, and is maintained by the ONV people. It's open source. You can download it. You can have a look at it. And I'm not going to talk too much about this because it's super duper interesting, but it's also an entire talk in and of itself. So I'll just leave it at that for now. The quality control, I guess I want to say a couple of words about that as well, because this is kind of one of the things that makes it a little bit tricky, this thing, because this is where biology means sort of computer science, right? So in computer science, we're used to everything being very discrete and sort of and no data loss, everything is sort of okay. And if it's a character is A, then we actually believe it is an A, and that's at least usually how it works in a text document, right? But the DNA sequencer is not perfect. So every few bases it gets something wrong, and the current raw read accuracy of the MinION, the machine that I showed you, is about Q23. So let's see what that means. the Q-score here is kind of like a logarithmic scale. It sort of works like a decibel scale. It turns an exponential scale into a linear scale, and it was invented to make it easier to talk about quality of base-called DNA sequences. So when I say that the raw read accuracy of the MinION hardware is about Q23, if you just plug it into the formulas here, you will basically get out 1 in 200. So 1 in 200 bases, it makes a mistake. So the accuracy is about sort of 99.5%. And this is not great, but due to redundancy, that's actually less of a problem that than you might think. So back to this figure here a little bit. It's usually the case that the sample that you put into the DNA sequencer contains many copies of the same DNA, because you usually take many cells and then you mix them together, and then those cells all have the same DNA, and then you get duplicates of the DNA. So the true DNA sequence is found here at the top, the sequence that you can see there. And then the sample is usually consisting of DNA then from thousands, if not hundreds of thousands of cells. And the DNA from these cells are mixed together, but they're also fragmented, as I told you earlier in the preparation process. So the sequencer essentially slurps through this mix of DNA fragments from thousands of cells, and what comes out at the other end, there are then millions of reads typically, each read from its own DNA fragment. And on the nanopore, these reads are usually then, as I perhaps mentioned earlier, hundreds to thousands of bases long, but for the illustration purposes, I've cut these down quite a bit, right? So once you've actually sequenced this, we get this sort of puzzle game at the bottom here, and then, okay, well, how to proceed from that then? If we do alignment, then we usually then well, let me rephrase that a little bit. If we know which species this came from, it came from a human, then we usually have access to what's called a reference genome. So for the most part, if you only have a couple of decimals, then all humans are the same DNA-wise, right? If you look at the DNA sequence in our bodies, they're remarkably similar, even though we are individual people. There are only very slight differences between us. So that's the idea there, that we can have sort of a more general reference genome, and the reference genome is going to be very much the same as whatever you sequence from a human. So you can use that reference genome to have an idea of perhaps where in the genome a specific fragment came from. So the true DNA sequence of this particular person might differ just in 3 or 4 places in this illustration here from the reference genome. And then since we know the reference genome, then it's going to be much easier to take the fragments from the puzzle game and just match them to the reference genome then. And then that gives us an approximation of how it would look like in the actual individual here. So then, for example, we could start out at the beginning here and sort of see, are there any fragments at the bottom here that matches the beginning of the reference genome? And sure, there are. Well, okay, so then we take those and we sort of remove them. And then we continue to the next one. And then here you will see that, okay, well, this individual has kind of a difference there in the third letter. The reference genome has a C and this person has a G. So we have to be a little bit lenient. We can't sort of match exactly, but we can have a sort of a Levenstein distance, or kind of like the fewest mutations that we need to do to get it there, and sort of use that as a ranking. And that's essentially what the alignment tries to do. Tries to match these things together, either to a reference genome or aligning these fragments themselves to each other. So, but if we wanted to do something more complicated, for example, if we didn't have a reference genome, then we usually get into a situation called the de novo assembly. In that case, we don't have the reference genome here in this figure. We only have the puzzle game at the bottom. And this is also a solvable problem, but it's much more computationally expensive, and it's not always perfectly solvable. There might be ambiguities here, and this is also actually a talk in and of itself. I'm not going to discuss it too much here, but I'm just going to give you a little bit of an intuition, at least the ones of you who did an algorithmics course at university level, or I guess any level, but there are 2 main kind of classes of approaches to this the problem is sort of the greedy approach, where you basically try to get to a local optimum as quickly as possible by sort of matching together 2 and 2 sequences and only doing the best ones at every local step. And there's a graph-based alternative that aims for the global optimum, and that's kind of where most of that, the software is going these days. It's a lot more computationally expensive, takes a lot of time to do, but gives you at least a global optimum result. So now that we know, at least at a high level, how DNA sequencing works, how and why could this be useful more concretely? Well, first off, it works in space, and anything that works in space is great. Obviously, this is a compulsory box to check before we can become an interplanetary species. But sort of more locally, it's also useful on planet Earth. For example, to take stock of how much life is left. By analyzing DNA from poop, for example, it's possible to identify genetic footprints of all animals in an area. This enables us to keep stock of the dwindling biodiversity. And as you know, there's a lot of fear-mongering in the ecosystem business these days. A lot of it is warranted, to be sure. But whenever somebody says this and that, many species are dying, now at least you can verify their claims yourself by sequencing some poop in your neighborhood. And going beyond sequencing, but still on the subject of poop, I'm happy to report that researchers have now used the nanopore to answer the age-old question that's been on everybody's mind. How old was this mouse that laid this poop in my kitchen? In some seriousness though, this is useful to aging researchers who don't want to take blood samples from their mice all the time. And for the last meat eater still in the room, it's now also possible to take a sample from the beef in the supermarket freezer and figure out where it came from. I spoke to a company a while back who cannot only say which country and region a specific meat came from, but also which farm and how old the animal was when it was slaughtered. Interesting enough, none of the producer organizations nor the free grazing / organic labeling organizations were too interested in this product, because there's a lot more organic meat being sold than what is being produced. However, the supermarkets who have to answer to their end customers directly, they're actually interested in this. And by the way, similar technology is also being worked on for plant-based products, but plant DNA is just a lot more complicated. So, if you have an inner genomic detective hiding inside you, just wanting to get out, where do you start? Well, you can, of course, shell out for a sequencer and then complete a wet lab course. And then you can start by sequencing some viruses and some bacteria or your own blood. That's easy enough that anybody can do it these days. But if you want to get your feet wet on the bioinformatics side without buying a sequencer, I would recommend you go and have a look at the Epitome lab tutorials and download the Epitome lab itself. It's basically a bunch of Docker containers with all the infrastructure inside. You can run on all 3 major operating systems. And helpfully, the Nanopore people have sequenced their chief technology officer, Clive, and the Cliveome, as it's called, the genome of Clive, is available publicly on S3 for everybody who wants to play with that. In conclusion, the democratizing price has now reached DNA sequencing. You can buy a brand new DNA sequencer for $1,000, and that gives you, if memory serves, also 2 free flow cells. That enables you to do DNA sequencing in the field as long as you have a reasonably powerful laptop. You can log along, and all the software necessary to interpret the output from the sequencer is open source, and most of it also is developed fully in the open. I included on the right-hand side here a graph just to show you the price improvements happening in this field. So when I said at the beginning that I think biology in many ways resembles the computing field in the 70s, 60s perhaps, then we're seeing these kinds of improvements over time that we also saw back in the day when Moore's law was invented. So that's really encouraging, I would say, sort of capacity-wise. And I think perhaps the most important thing of this talk in total is that it's now possible from a piece of poop for you yourself to determine someone's age, gender, and even eye color if you should so desire. So with that, I'll leave you to have fun and drink a lot of genetically modified beer. Thank you. Hi, I'm Karl Trygve Kalleberg, one of the Hackeriet regulars. Today I'm going to venture out on a little bit of a limb and talk.
Once is probably enough, don't you think? So that was a recording. Yes, we fooled you, and here's the real deal. Hello, girl. Oh, we can't hear you. Do you have sound?
I was just saying this is also a recording.
This is also a recording. Yeah, nice. It's now possible to ask questions through this modern piece of technology, and I'll have to be the proxy for this because the microphone he hears is in my laptop. I'm going to start by asking, where is this going in the future? Like, what kind of new life will we be seeing in 10 years based on this? Do you have some ideas?
Yeah, that's a great question, right? So, I mean, in 10 years' time, I think there'll be a lot more sequencing going on for the kicks and giggles in primary school, universities, any kind of biology course. And I think the next generation of this particular technology, they want to make the sample preparation also inside the same machine. So you should be able to just put the sample directly into it. But I think sort of in 20 to 30 years' time, perhaps it's going to be so pervasive, so ubiquitous that everybody will be sequenced all the time. So if you go to a public toilet, for example, and pee, your pee will be sequenced, right? And then there will be a warning coming up then saying, well, you seem to have prostate cancer, you should go check with your doctor. Goodbye.
All right, yeah, that's a feature, isn't it? Did you have a question? Yeah.
So
For the assembler.
what's the average length of or the mean length of the read, and
What's the mean length of the read?
And the variability?
And the variability.
All right, yeah. So that depends a lot on where the sample comes from, biologically speaking. So which DNA you're working with, for example. So if you think about it, and you take it from a bacteria or a virus, many of them have rather small genomes. If you take it from a human, which is a relatively long genome, then the fragments become bigger. But it depends there also on which type of sample from a human you take it from. If you take it, say, from the blood, like the DNA in your white blood cells, for example, versus taking it from a tumor biopsy. So I don't think there's an easy easy answer to give you there. I mean, generally, sort of the best I can give you is sort of from the hundreds to the thousands, and then of course there is even a what's it called challenge of getting to sequence the longest reads, and I think those are now almost in the millions in the best of scenarios.
More updates on that? More?
So the fragmentation obviously depends on the preparation.
The fragmentation depends obviously on the preparation.
So I was trying to figure that part of, is there something that can be done about
is there something that can be done about the preparation to increase the read sizes?
Yeah.
And
Yeah.
Let me finish. And then because what I was originally trying to figure out is if the read sizes are too small, then the problem becomes unsolvable.
if the read sizes are too small, the problem is unsolvable. You
Yeah.
conclude.
Yeah, I think that's a very valid conclusion, to be honest, right? So the answer is yes. There are various ways to do the chemistry and the preparation to preserve the DNA as well as possible. And there is even a whole field called single cell genome sequencing. And that's when you try to sequence the genome from just a single cell. And of course, then you'd like to have the DNA is as great quality as possible. And there are multiple protocols that have been published by various research groups sort of every month. It seems like somebody tries to improve how to do that. So yeah, that's a combination typically of both chemistry preparation, sample selection, and also the, of course, then a couple of downstream analysis parts. But I would like to say and also point out that remember at the very beginning of the talk, I showed the sort of mainframe alternative, the Illumina sequencer. And that's honestly
The next
what's
generation one.
yeah, the NGS one, yeah, indeed. So that's the workhorse of today. That's if you look by volume, that's the kind of machine, or that family of machines is the ones that sequence the most genomes in the world, and they have a restriction that they're only able to sequence about 150 base pairs. So that's why this sort of sequencer I'm talking about now is called a long read sequencer, and the other one is called a short read sequencer. And then, of course, the assembly for the short read is a lot more tricky. And it turns out then that in our genome and in the genomes of many animals and species, there are repeated sort of sections that are so long that you don't really know with a short sequencer that you get it right, because it's long. The repetition is longer than 150 base pairs, and you don't. Then it's very difficult to know if you got the number of repetitions correctly. So those are some of the sort of advantages of the sort of the Nanopore sequencer, is that they can resolve some of those repeated sequences in a much better way than the Illumina machine can, for example.
Yep.
You said that you don't always need to assemble the whole genome.
You said you don't always need to assemble a whole genome.
And you can just
Yeah.
And
what do you do that, or do you know what part of the genome you're actually looking at when you have just a line?
do you know what part of the genome you're actually looking at when you have
Just a line.
just
Not
aligned and not fully assembled?
Right. Yeah, indeed. So depends on the application, right? But if you're say into cancer, I guess is sort of a typical example, right? Because various cancers are usually many cancers are dependent on specific mutations at specific sites in your genome. So you don't really need to assemble all of the genome in order to know if you have a mutation in one specific gene. You only need to put together the DNA sequences for that region that you're interested in. And that is relevant for cancer detection, obviously, but also various kinds of research applications where you only look at the mutations in certain regions of the genome. And then there's a chemistry trick that you can do along the sample processing, where you basically select chemically the regions that you're interested in. You protect those regions, and then you have a way to ingest or sort of just dissolve all the other DNA that's not protected, and then you unprotect the region, and then you sequence that. That's called targeted sequencing. So the whole point about that is basically throw away all the DNA that's not what you want to look at, if you know exactly which subset you want, and then leave the rest for sequencing. There's, I mean, I guess I should throw in another hack there that's very specific for targeted sequencing for the nanopore. You can actually run the pore in reverse on a per pore basis. So what you can do, you can let the DNA start being sequenced, you do base calling in real time, so you know as each base goes through. That's slightly less accurate, but it can be done. And then if you don't like what you see, if you see that this is a sequence that cannot be what you're looking for, you can basically run it in reverse, and you can eject that DNA sequence, and then you can wait for another one to show up. I mean, which one shows up is just a matter of luck. This is chemistry or sort of biology after all. But then you can try it again, and if it's the same thing, you can eject it. But in that way, you can avoid sequencing things that you don't want. This is called adaptive sequencing. And on the topic of that, I mean, this is where it gets cool, right? It turns out that if you run the DNA in reverse through this core, you also get the signal. And that signal has different error properties than the ones that you run in the right way. So now they're experimenting with running the DNA back and forth through the core multiple times to improve the signal and then improve the accuracy of the read.
All right. Yeah, analog is fun, isn't it?
Yeah, it is very analog. Right.
All right. Well, that was all the questions we had, as far as I can see. Yeah. Thanks a lot.
Good. Yeah.
Guess we're all gonna have fun with Wetworks now in the future. Now. Thanks.
Bye.
Have a good day. Bye.